Thank you very much for this introduction. Yeah, after the talk of Pietro, you would almost say there's nothing necessary anymore. This is like a success story, but I'll try to convince you that indeed new treatments are needed for hepatitis B, and those are being developed. A lot of companies now are reinvesting their research dollars from hepatitis C to hepatitis B because we're doing even better on hepatitis C, and we really don't have a good functional cure for hepatitis B. So the key consideration for the current treatment option in hepatitis B is that indeed all the HBV nucleotides are generally well tolerated, but low rates of discontinuation. They have to be given long term for that reason. And PEG interferon monotherapy is finite, but only effective in a subgroup of patients, and its use is very limited due to toxicity. It's really not a pleasant drug to use, even not for a couple of months. So why is finite treatment a goal for HBV treatment, or for any kind of disease, basically, most probably? Younger patients might find it hard to accept lifelong therapy. We think they do, but they're not always. There's a problem with long-term adherence. A lot of patients say they'll take their drugs, but they don't do it as well as we think. We have young women who might become pregnant, and then we sometimes have to interrupt their treatment if we don't know what it does to the child. There are definitely cost savings to the healthcare system, and actually a lot of patients are even reluctant to start on treatment if you tell them that it might be given lifelong. So if you look at the current treatment paradigm, it's indefinite treatment, poor off-treatment response. It reduces overall mortality, I think. It reduces but not eliminate the risk of HCC. And again, Henry will tell you more about that. In the large majority of patients, I think that NUCs will not lead to cure or even functional cure. And the new paradigm would truly be to give a finite treatment, no increased risk for mortality of HCC. So I structured my talk in two different parts. I'll start off with strategies with licensed medications, shortly touching upon the combination therapy of PEG interferon and nucleoside analogs, and then I'll move on with unlicensed and newer medication. So the saga of nucleoside analog and PEG interferon treatment has been ongoing for, I think, a decade now, but has not ceased. And that's because the drugs are very, you would say, theoretically very complementary to each other. So if you give an antiviral in combination with an immune-modifying drug, so theoretically you would say you would have a very, very good combination. And several studies have been done, and several studies are still ongoing in this field. And thus far, the success rate of combination of PEG interferon and nucleoside analogs is not surpassing the effect of PEG interferon alone. So what you sometimes see, or at least our experience in the past, is that during treatment you see a better response rate of both of the combination group in comparison to PEG interferon alone. But once you stop treatment, you kind of lose the effect of the nucleoside analogs, and you see seroreversions or reactivation of the disease in the combination group, likely due to nucleoside analog-induced E seroconversions or E losses. And if you compare the groups, you end up with the same amount of response in PEG interferon monotherapy as compared to the combination of PEG interferon and nucleoside analogs. So we have been looking on more clever ways to combine these two drugs. And one way is to start off with a nucleoside analog. And we know from several studies, particularly from some Italian studies out of the group of Ferrari, that if you do give an antiviral, that you do get a partial immune restoration after viral load reduction. And it happens with all nucleoside analogs. It really doesn't matter whether you're giving entecavir or tenofovir. We do see that in all different levels, both in the innate and the adaptive immune response. However, in clinical practice, that's not enough, because we know that once we stop these drugs, that most of our patients relapse. So one theory could be is to use nucleoside analogs as a priming therapy to get that partial immune restoration, and then kick in with PEG interferon as an immune-modifying drug. So this is what we did recently in this particular study, and there are others who've done it as well. This is a study that's just been published in Hepatology, where we started off with entecavir monotherapy, and then after 6 months of treatment, so 24 weeks, we randomized towards add-on with PEG interferon, or just continue with the entecavir. And you do see quite a profound difference here. In the add-on group, we saw significantly higher rate of response as E seroconversion and low HBV DNA defined. More decline in S antigen, E antigen, HBV DNA, and also more sustained response after E antigen seroconversion than in the entecavir only group. So I do think that a PEG interferon-induced E seroconversion is immunologically different than a nuke-induced E seroconversion, and we do tend to see much less relapses than in the nucleoside analog-induced E seroconversions. However, this study was, I would say, rightly criticized by the fact that there's no interferon-only group in there, and we still should, I think, compare these combination options to interferon only. So let's move on to the strategies with unlicensed medication. And there you start to look at the life cycle of the virus, and there's a lot of action ongoing in the development of the different drugs. And I'll highlight here in the yellow boxes here what is currently done, and many of these drugs are still in very early development, but in the different stages of the life cycle of the viruses, as well as the immunological control, many companies are working to develop new drugs in this area, which is a very good thing if you've been interested in hepatitis B like myself for 20 years and not have been approached for the last 10 years because everyone was interested in hepatitis C. So I'll talk about a virological approach as well as an immunological approach, and let's start with the virology part. One drug already mentioned by Ray Shinazi, and I look. This presentation is from a clinical perspective, so as a clinician, I look which drugs are approaching the clinic in any way. And this is one of the drugs which is farthest along, I would say. It's a kind of super tenofovir, tenofovir alafenamide fumarate. And it has an improved stability in plasma, and it would have enhanced delivery of the active form of tenofovir to the hepatocytes. And for that reason, you can give it in a much lower dose with even less toxicity than tenofovir has at this moment. It's a once-daily oral single regimen, which has already been used in HIV. 25 milligrams of TAF was similar in antiviral activity, but reduces systemic exposure to less than 8% of the exposure generated by 300 milligram of tenofovir, the usual dose. So it's probably less nephrotoxic than tenofovir. It's also beneficial in HBV and HIV co-infected patients, and the phase 3 study is ongoing, which is depicted here, where people are randomized towards tenofovir and tenofovir alafenamide with the primary endpoint of HBV DNA undetectability and secondary endpoint, particularly on toxicity, because if we have to give these drugs for decades, they have to be very, very safe. And even 7, 8, 9, or 10 years is not enough to detect that. Does this drug give more functional cure of hepatitis B? I think the answer is no. It's another nucleotide analog in this case, which is probably potentially better than the ones we have. But from a theoretical viewpoint, I would not see how this drug could lead to more S loss or functional cure of disease. Then we have besofovir, which is an acyclic nucleotide phosphonate, effective for naive and limb-resistant patients. A multi-center trial was published in Gut last year, where it was found to be non-inferior to the usual dose of entecavir, no resistant mutations or renal toxicity, but within this group of besofovir-treated patients, oral carnitine supplementation was necessary, basically. So that's not something you want. And I mean, we really have to develop very good drugs now, considering the fact that entecavir and tenofovir are so good and so safe. So we'll see how this will move on. So let's move on to other concepts of treatment. And I think an important discovery for this was the discovery of the NTCP receptor done in China, which is kind of the functional receptor of hepatitis B virus and really opens the door for high throughput drug screening, as well as a new target basically for treatment. So this is a receptor binding region of the PreS1, specifically interacts with sodium taurocholate, co-transporting polypeptide, and it's a multi-transmembrane transporter. It's predominantly expressed in the liver. And one of the drugs targeting this receptor is Merclodex, which is a chemically synthesized lipopeptide derived from the envelope of the HBV virus, which would block infection in cell culture. It does do it in, or there's evidence that in HepG2 and HUA7 cells that this is happening. In animal models, it prevented the establishment of de novo HBV infection. It strongly disseminated HBV dissemination and the accumulation of cccDNA in mice. And a phase 2 study is ongoing, and I think next week in Boston we'll hear the first results on that rather new approach. Then there is RNA interference. HBV is susceptible to RNA interference because it replicates via an RNA intermediate. Small interfering RNA therapeutics have the capacity to knock down production of hepatitis B genes and thereby significantly decrease the number of infectious particles. And you would hope that this reduction in virus and antigen will mount an immune response which will take care of the virus. I do not expect that this approach will get rid of hepatitis B by itself, but you would hope that with the decline, particularly in S antigen, because most of these RNA interferers are aimed at antigens, particularly the S antigen, and you would hope that decline of S antigen would result in cure of disease. And that's still questionable, because the question is, what is the chicken and the egg, basically? Now, as a result of an effective immune response, we do see S seroconversion, but the question is, if you kind of artificially bring down the S antigen, like you might do with these compounds, does that result in the same type of cure? That's very questionable, and the only way to sort this out is really in humans, basically. It's very difficult to do these studies in animal models. Antisense oligonucleotides, there's also several companies working in that field within RNA interference, and phase 1 randomized placebo-controlled trials are about to be started. These are drugs which are still in very early development. There's always a risk of, there's a profound risk of potential toxicity, so we don't have to cheer at all with these type of compounds. We really have to wait the results. So if you look at the different strategies, and I didn't bring them all to you, but we have entry inhibitors, we have assembly effectors, we have RNA interference, we have new nucleoside analogs, we have HBs antigen release inhibitors, and also drugs which target CCC degradation with upregulation of ApoBec A and B. And we hope they'll be successful. It will be pretty difficult, but we're by far not there yet. And the road to CCC degradation will be pretty difficult, I think. So let's move to the immunological field. And I'll talk about PEG interferon lambda, about toll-like receptor agonist, therapeutic vaccination, and PD-1, PD-L1 blocking. We had hoped that PEG interferon lambda could replace PEG interferon alpha, but it didn't do it for hepatitis C because it was surpassed by many more, let's say, more effective drugs, antivirals. And still, it's an interesting compound to test for hepatitis B, and it was tested under leadership of Henry Chan. But unfortunately, it was not very effective. The problem with PEG interferon lambda and hepatitis B that not a good dose-finding study has been done. Still, there were good reasons to compare 180 micrograms of PEG interferon alpha to 180 micrograms of PEG interferon lambda. And during treatment, so this is 1 year of treatment, a kind of similar response was seen as E seroconversion with the endpoint. But then off treatment, there was more relapse actually in the PEC interferon lambda group, whereas in the PEC interferon alpha group, response increased due to probably immunological carryover effect of PEC interferon. Why this happened, we really don't know. But with these results, BMS, I think, has sort of unfortunately decided to stop further development largely of this compound in the field of hepatitis B. What about toll-like receptor? There are several companies working on this, and TLR7 is a pattern recognition receptor in the compartment of endolysosomal compartment of plasmacytoid dendritic cells, so PDCs and B cells. And agonism would induce an antiviral response via innate immune activation. The compound called GS9620 is now being tested in combination with oral antivirals. And it was potent in several animal models. And also there was a decline seen both in HBV DNA and S antigen in chimpanzees. And thus far it seems to be well tolerated with significant dose-dependent effect of immune stimulating genes in patients. We'll see how things are moving along. Again, the difficulty of testing these drugs is that you do want to stimulate the immune system to get immune control over the virus. But if you overstimulate the immune system, you might end up with nasty flares in the field of hepatitis B. And we don't want that. We don't want to get relatively healthy patients into a state of liver failure and have to call the transplant surgeon. That's not what we would be very happy with. So let's move on to the field of therapeutic vaccination, and I'll just pick out the two vaccines which have been developed recently or which are in development. One is called GS4774. It's a Tarmogen. It's made from genetically modified yeast that expressed one or more disease-associated antigens, so you can pack it with different epitopes, and then you hope that it activates T cells and specifically targets the hepatitis B virus. Thus far, it's been well tolerated, and it's currently being assessed by Gilead in patients, again, who are virally suppressed, and on top of that, get this particular compound. So most of these studies are done in patients who are already on tenofovir and entecavir, and then with these immune modulating compounds, you hope to tip the balance in such a way that these patients will get S antigen seroconversion. Another reason to do that, basically, in the setting of virally suppressed patients, is that they hope to dampen the potentially side effects of these compounds. Another therapeutic vaccine is this one coming out of France, TG1050, based on a non-replicative E1 and E3 deleted human adenovirus. It contains the core as well as polymerase as well as the S domain. It's genotype D sequence, and it's capable of inducing multispecific sustained cross-reactive T cell responses, and the first in-human trial is ongoing this year as well. And I think the concept of therapeutic vaccination is a very good one, but we've been working on this already for probably 15, 20, 25 years, and it thus far has not worked very well. And the reason might very well be that these patients that we try to treat have been exposed to the virus and to the viral antigens not for years but for decades, so are completely tolerant to some of the stimulation, and for that reason it's very difficult to achieve a good immune response. Both of the compounds that I just discussed have their problems. And regarding the Tarmogen, it's a vaccine which is not delivering the polymerase antigen. The X antigen is poorly immunogenic. It's likely an issue related to antiviral neutralizing immunity. And the technology didn't demonstrate a lot of effect in hepatitis C. The transgene compound has the problem that adenovirus have limited used due to pre-existing immunity, so you can have antibodies to the adenovirus, and it's likely restricted to specific genotype coverage. The last, let's say, therapeutic mode that I would like to discuss is this one, where also several companies are working on, which is the PD-1, PD-L1 blockage. So there's an inability to eliminate chronic hepatitis B that has been attributed to the expression of these programmed death 1 and programmed death ligand 1 on viral antigen-specific T cells and antigen-presenting cells. And the thought is that if you would block the interaction between PD-1 and PD-L1, it at least in vitro reverses the exhausted cytokine production and proliferates hepatitis B specific T cells. So you can revitalize your hepatitis B specific T cells, and you would hope for a good immune response. This concept is used in the setting of oncology and HIV, and it's now being tested in hepatitis B as well. And it's thus far completely preclinical as far as I know. So if you summarize all of these different compounds and different modes of actions and the phases where these trials are in, I think we're moving into a very exciting area. I think it will be very difficult to get full cure of disease or even functional cure of disease, and it will likely very much dependent also on some host factors. And we didn't have any good, I think it's very important, for instance, that we would have good data, GWAS data on interferon responders in the setting of hepatitis B, because I think that several patients are just more likely to respond to any kind of treatment than others. And we might end up with a curative regimen which is a combination of different approaches. So there's the antiviral agents which can prevent viral spread. There's the immune activators to activate antiviral immunity and relieve repression of the system. We can work on cccDNA and try to inhibit that or deplete that, and on HBV antigens. And very often, if I talk to my virologic or immunologic colleagues, they say, well, let's just combine all of these. But as a clinician, you know that it's not that easy, and that it's sometimes also not without risk to combine these different agents in our patients. So it will take a while before we can do that. But I think that truly this is the way forward. So to conclude, Mr. Chairman, ladies and gentlemen, I think now that we're very well able to suppress hepatitis B, our next challenge is really to get cure of disease. I mean, hepatitis C is a curable disease. Hepatitis B thus far is a treatable disease, and we have to make it a curable disease as well. And the best endpoint of that is probably S seroconversion, S antigen being indicative of transcriptionally active cccDNA, we think. So it's the best endpoint we have, but we just discussed that it's still, as a biomarker, we're looking kind of a black box, and we really don't know what's happening in the hepatocyte exactly. I think we have to do more studies of the combination of our most potent nucleoside analogs and PEG interferon in different regimens. HBV entry blockers, antisense therapy is promising, but in very early development. Direct CCC inhibition may be needed, but is difficult to reach and will not be the holy grail, in my view. And again, you need a certain amount of immune control to suppress, and otherwise one patient or one cell infected with cccDNA would be enough to get the infection started again. PEG interferon lambda is disappointing thus far, and I don't think we'll hear too much about it in the future. And different immune-modifying agents with hopefully limited systemic effect can potentially be combined with antiviral agents. Thank you very much for your attention.